Okay,
Excellent.
there we go.
Oh, nice.
All right, so I'll put it on my website for a number of people that. Okay, drop that. So today, we're going to do ancient DNA and what's happened in the last 25 years, basically, a whole new revolutionary way to do paleontology, basically. And the guy who is absolutely responsible for it is Pavo, who is basically the grandfather of evolutionary genetics, and was the one that started believing the idea when he presented it to his PhD thesis supervisor. He laughed him out of the room, basically, that you could get DNA from dead things, basically. So, and this is the classic finding that one of the first, that basically Neanderthals interbred with modern humans. Originally, the concept that around 54,000 years ago, and that it was possible to prove this scientifically. His first book from 2014, his only book so far, is Neanderthal Man in Search of the Lost Genomes. It's an interesting read, both the autobiographical and research data. And last year, he deservedly got the Nobel Prize for medicine and physiology, basically. Well deserved. So I'm going to do a quick update on genetics because it goes through this talk. Basically, we have 3 billion base pairs in our genome in each of our cell nuclei. Currently, the thought is that there are about 19 or 20,000 genes that are code for creating protein. There are 23 chromosomes, and basically the genome works by these four ingredients, A, T, C, and G, combining along the genetic ladder, basically. Between any two of us, the DNA is 99.9% identical. Okay, so only 0.1% is different between any two of us. Every new baby that gets born ends up having instantaneously 100 to 200 new mutations in those 3 billion base pairs. And between any two humans, a mutation occurs every 1,200 bases, as opposed to chimps, where it's every 100 bases. And each of us has two genetic strands from each parent. So in effect, we have 6.4 billion nucleotides combos there.
Hey, Charlie?
Yes.
What's the significance of having one every 1,200 versus having one every 100?
You've got more.
Oh, you got more. Okay.
We get more mutations, basically. So
is that a plus or a minus?
It's just
yes,
the way evolution worked.
Oh, okay.
So in the 3 million base pair differences between any two people, so the 3 million little one of those ACGT differences between us. And so a base pair is basically any two of those ACGT combinations, and they form the rung of the DNA ladder. A genetic loci is a specific fixed position on a chromosome where a particular gene is located. An allele is one or two or more alternative forms of a gene that arise by mutation, but they're found in the same place on a chromosome. So as an example, the APO4 gene that can give you Alzheimer's is an example of an allele where you can have 0, 1, or 2 of them. And recombination is that during meiosis, when sperm meets egg, the DNA gets totally scrambled, broken up, and recombined in that process to produce a whole new 3 billion set. Statistically, this is a weird one. There's a 1 in 8 million chance that you can be genetically totally unrelated to one of your 4 grandparents because of recombination. If it happens in a weird way that one of them, the genes from one of them are totally excluded in the recombination just by chance, okay? So you can, it's one of the reasons why you get more genes from one parent than from another parent. Typically, I was surprised when I first saw on my mother, my brother did, and I have done our DNA from a couple of companies out of curiosity. And my brother had 52% from my mother, and I only have 50%. Okay, so recombination.
So Charlie, is that where that saying, "You take after your mother or father" comes from?
That's the basis. Okay. So the base pair are these little guys along the chromosome backbone, where they can combine in these particular ways, basically. So they're made up of thymine, adenine, cytosine, and guanine, okay, are the 4. Now, your mitochondria, you have thousands of them in every cell. We basically absorbed these sometime back 1 billion years ago, sometime way back then. But they're the powerhouse of cells. They produce the energy that's needed. But inside of them, you actually have a DNA strand that consists of exactly 16,569 base pairs that constitute 37 genes in the mitochondria. And the importance of the mitochondria is that these are maternally inherited. It goes from grandmother to mother to daughter to daughter to daughter to daughter. But note that these have far less information DNA-wise than the nuclear DNA, which has all of the 3 billion, okay? The Y chromosome is used to trace paternal, here, I'm just moving something, ancestry, and the mitochondrial traces maternal ancestry. They can now compare computerized sequences of all 3 billion base pairs for between any 2 of us. Okay, and this is what it looks like when they in effect print it out, and they can tell by tagging each of these with a fluorescent tag. The computer can then read the different color and then put in the correct A, C, G, or T into that particular place. So here, everything's the same except for these 2, which are different. And again, there's one difference in every 1,200 letters or so between any 2 of us. Now, genes are the DNA that code for proteins, which is what builds everything in our body. And they're along the 3 billion strand. They can be very small to millions of base pairs long. And then there is the non-coding DNA, which is 99% almost of the entire genome that don't code for making a protein. And originally, this region was entitled junk DNA because they thought it didn't do anything. Well, they've been disabused of that belief because it turns out about 7%, if I remember correctly, of these non-coding regions actually control the expression of a gene, particularly turning them off, okay? So that they're hugely important, it turns out, just as much as a gene, they just do something different, okay? So old junk DNA, now regulatory DNA, and they carry instructions for gene regulation, basically, and they can activate or inhibit a particular gene's activity, okay? So there are about 1 to 2 to 3% that are the 19,000 genes. The rest is non-coding. Before the Human Genome Project, there was an idea that we had 100 to 250,000 genes based on the number of proteins we make. They thought it was one gene, one protein. Now we know that genes make an unknown number of proteins. Only about 19,000 of them have been confirmed, but there are a lot of other ones that we don't know the connection yet. And trying to get my clicker to work here. Okay. So DNA outside protein producing areas are really important, not junk. 5 to 10% of the human genome is considered to be highly conserved across all mammals, and it implies that they're required for survival. You don't want to mess with those, and a mutation in those areas can kill you, basically. So you want to protect those. And only about 1 to 2% code for proteins. And a little over 1% accounts for all the proteins that we create, basically. The other 7% of the regulatory areas are involved in switching on and off the genes that encode the proteins. And most mammals have the same setup, basically, in terms of non-coding versus coding. And interestingly, the amount of research now is much more about the non-protein coding regions than they are about the genes, okay? 30,000 papers per year are looking at trying to link a particular base pair and what traits they control in human. The big secret, Pablo commented, was that we know almost nothing about which areas of the genome control what in terms of our phenotype, basically. But still, much of the DNA is in fact not well known. Now, DNA can mutate at random due to ultraviolet radiation, any form of radiation, including X-rays that you get at your dentist, and potentially errors during cell division that normally is corrected for by your DNA, but sometimes it misses. And having too many bad mutations will basically kill you. So the conserved regions are really important. A haplotype is a combination of alleles at different chromosome regions that are closely linked and tend to be inherited as a group together. And this can happen both in the mitochondrial and the nuclear DNA. So it turns out that mitochondrial Eve, who we'll talk about, is haplogroup L, okay? And so she was, wherever in Africa she was the original mitochondrial ancestor of us all, basically. And since then, as we left Africa, we mutated, and eventually, as an example, one thing they know about Native Americans is that they represent only about 5 haplogroup types. And it's the way they prove that they were connected to the group up in Siberia as their point of origin, but different haplogroups in different regions. Now, chimps have vastly more genetic variation than modern humans do in all of the studies. And secondly, African Homo sapiens has the greatest genetic variation on the planet today. Okay, so that was the original founder group, Africa. And as we left in migration, each group that left had less of the original amount of DNA. That's an important point. And as a consequence, of course, it means that we are all African by DNA, okay? Even white nationalists, okay? So ancient DNA is a field of molecular evolutionary biology that uses DNA sequence data recovered from poorly preserved organisms that may have died hundreds to potentially millions of years ago. And we now can extract this degraded DNA from not fully fossilized fossils, okay? That's important because DNA is not fossil. It's organic material that's still organic and is not fully fossilized. So you can't get DNA from a fully fossilized bone, as an example. And the current estimate of preservation potential for ancient DNA is anywhere from 10,000 years ago to 2 million years ago or so, as we'll see. It's been an amazing technological revolution that has basically given us the ability to do this. And as a consequence, we've discovered that there have been interactions between archaic, like Neanderthal, and modern humans, modern human population dynamics and migrations. As an example, what happened in Europe 40,000 years ago, and the settlement history of regions of the world. We'll talk about India as an example. In 2001, the draft sequence of the human genome was published, and it's updatable, and it has been updated, and it's now used as the reference genome anytime you want to compare a new ancient DNA finding to this reference genome to see whether it's different than us. Now, there are actually three ancient biomolecules. DNA is one of them, nucleic acid, but proteins and lipids are also possible ways to look at ancient material. DNA can show evolutionary processes at the highest resolution. You get the most data. Proteins and lipids can be gotten from much, much older than ancient DNA, but you don't get as much information from it because they're different than 3 billion number. So we've been able to kind of reconstruct both speciation, migration, and admixture events from lots of different extinct species. So since the advent of this, we can do small mitochondrial and nuclear DNA fragments retrieved from a single species, multiple species, full genomic sequence of one or several specimens, to single nucleotide polymorphism capture-based population genomics, and a sequence called shotgun sequencing that can include hundreds of people at a time, okay? So the field just keeps expanding. And how it's been applied in evolutionary biology is we can now understand human evolution based on this technology. And they've been doing a lot of it in terms of ancient hominins up to, in Europe, typically around 50,000 years or so. Now, we mentioned that DNA degrades and it survives best in cold. So permafrost areas are where you want to find them. I'm still hoping that eventually we will find a Neanderthal in permafrost so that we can prove that they wore trousers, as an example. It's the only way we're going to find out what their clothing was like. By contrast, tandem mass spectrometry can be used as the technique to actually look at 1.5 million year old collagen proteins in a bone or a tooth, etc. And so you can use protein analysis where you can't use DNA because DNA may not last that long. And so you basically find a collagen and put it through mass spectrometry, and then you can basically, it gives you a spectrum that identifies the species. So this happens to be the cow spectrum in a mass spectroscopy machine. This is the human spectrum. So you can identify the proteins that belong only to hominidae, okay, us. And this is the technique that they used on this incredibly little bone. It's less than sort of, I think, 1 or 2 inches, to prove in Denisova Cave, Siberia, that this was a girl who had a Denisovan and Neanderthal set of parents based on proteins. Okay, we'll talk about her. So she's the daughter of a Neanderthal mother and a Denisovan father. Okay, so that's the level, and that's how small this little piece was that they got the protein from, not the DNA. And that the headlines, of course, were hybrid Neanderthal love child found in cave. Prehistoric humans did hanky-panky. Shows study. Okay, you get some wonderful headlines out of these findings. Okay. So again, ancient DNA, not in things that have been completely fossilized because
Say, Charlie?
Yes, Dennis.
How did she get the name Denny instead of Denise? Is it they named
the guys at Denisova Cave.
Oh, Denisova Cave. That's how.
Named it after the cave. And the cave was named after a famous hermit, a Christian hermit from long ago who lived in that cave. So that's how the Russians named it. Okay. Now, one quick comment about molecular clocks, because you read a lot about this, and that is, this is a technique that uses the mutation rate of biomolecules to deduce the time in prehistory when 2 or more life forms diverged from each other. So it can tell you how far back 2 things speciated separately, okay? And they can use the nucleotide sequences from DNA, RNA, or amino acids to start the clock ticking on this. And it turns out that neutral mutation, random changes in DNA, occur at a constant rate in all mammals, as an example, okay? And you can use this mutation rate to check further back to see how many. The more mutations you find, the longer back it is. The less mutation, the shorter the time period. So you can compare the number of mutations in 2 species to arrive at the time of the species diversions, or the last common ancestor. And this is how they get the estimates for, as an example, that we left Africa 60K ago, is based on mutation rates. Mitochondrial was the one they used for that one. So you can also do phylogenetics based on this. So you can create a tree based on these mutation rate data. Okay, so that's pretty much it. So more mutations, longer timeline. the last common ancestor. And if you look at your mother and your father, they can have very few differences, which means that they had a common, the two of them had a common ancestor 50,000 years ago, or they can have a lot of mutations showing that in fact the last common ancestor was 1 million years ago. Okay, so it's a great biological tool for telling time.
So Charlie, in the academy, then the display that shows a 70,000 year divergence with the DNA more homogeneous before, is this how they determined it? By looking at
mutation rate.
Okay, so basically they looked at what? They looked at tissue from before then, and they found the rate higher than it is now?
Well, they look at modern humans and then start checking their mutation rates and what it tells you about how far back the thing is that you're looking for. And it's also the way, we'll talk about this later, about how they determined how many people were at that bottleneck period. Okay, we'll come back to it.
Okay, great. Yeah.
Okay.
I ask that all the time, so that's great.
Yeah. So you can get ancient biomolecules, DNA, et cetera, from practically anything. Okay, eggshells, snail shells, coprolites, poop, dental calculus on your teeth, keratinaceous materials like hair, claws, feathers, ancient plants, seeds, fruit, wood, and as you'll see, mummies, organisms in amber, plant materials in ancient tombs, bacteria, bones, pages in a book. They proved, as an example, that nuns back in medieval time were also doing manuscript writing as opposed to just monks, based on what they found of the DNA in parchment, basically. Now, the most recent discovery, importantly, is you can get DNA simply from dirt in a cave. So if a Neanderthal pooped or peed in a cave, they've left their DNA. And you can grab a bunch of dirt and pull out any species that was in that cave, independent of any bones or fossils. Okay? They've even figured out in naked mole rat tunnels that they can get the DNA from the air to show interesting facts about those little creatures, okay? They discovered a piece of tar that was dated 8 to 10,000 years ago that had been that was chewed by an 8-year-old girl that had brown eyes. Okay, just from the DNA that's left on a piece of 10,000-year-old tar. Okay, so hair, plants, coprolites, any tobacco lump that you chewed, dental calculus, ice cores, parchments, clothing. And most famously, this is the skull cap of Neanderthal 1, who was found in 1856. And they didn't use the skull cap, they used one of the femurs from this skeleton to come up with the first Neanderthal DNA. The first Denisovan DNA came from a little piece of pinky finger bone from Denisova Cave. The oldest Neanderthal they showed was 430,000 years old and from a femur at the Cima de los Huesos in Atapuerco, Spain. This is the horse bone that they showed was 780,000-year-olds in the Yukon. And this is Utsishmim, the guy from Siberia that they dated to 45K based on DNA they got from his thigh bone as examples of the material they can use. It turns out that the petrous bone, which encapsulates your 3 little auditory bones, turns out to be the densest bone in the human body. And this has been the great bonanza in ancient DNA for hominids, that they can find incredibly old data from this particular bone. And Pablo's group at Leipzig were the group that discovered this. So it's the best source of paleo DNA. It's only about an inch long, but basically because this is such a delicate region to let those bones, move around, that this is the protective area that turns out to be spectacular for DNA. And David Bowie said, "I don't know where I'm going from here, but I promise it won't be boring." And that's been the nature of the discoveries, basically, going all the way to recently sediment DNA, basically. So the two main complications for studying this stuff is that as ancient DNA, dead DNA from a dead creature, that molecular damage starts off almost immediately. And then you start getting contamination from other creatures on the thing you're looking at, okay? So these two are the big things that they had to conquer in order to finally achieve what we've been able to do. Back in the 1990s, just as Spielberg's Jurassic Park came out, the first peer-reviewed publications claimed that they had dinosaur DNA, okay? And then the question, of course, was it possible to resurrect dinosaurs, et cetera, started popping up. In the New York Post, mad scientist clones dinosaur for defense purposes, okay, popped up. And if you want to read about this whole time period, there's a book called Ancient DNA by Elizabeth James that looks at all the interplay between the movies, the publication of the journal articles, et cetera. They played off of each other, basically. The question, can you get genes from dinosaurs saved in amber? And the problem is, of course, is DNA starts to increasingly degrade the longer it's been in the ground. And there were a bunch of papers that were peer-reviewed that claimed that they had discovered DNA from dinosaurs. And all of them were absolutely wrong, but they've never been retracted, okay? DNA begins as one incredibly long, 3 billion base pair long strand. Once you die, well, actually, before you die, sunshine, ultraviolet radiation will break down DNA in your skin, but there are proofreading enzymes that correct for it, basically. And once death occurs, DNA begins to degrade, breaks down to ever smaller and smaller and smaller fragments. Then in the environment, UV radiation, oxygen, particularly water, enzymes in your gut, microorganisms in the soil, all begin degrading the DNA. So all ancient DNA is heavily fragmented and chemically modified, okay? And the concentration and the mean fragment length declines exponentially with age, which is why we've never found any DNA from dinosaurs, okay? And you can't taxonomically identify due to the absence, partially of the fact that for many creatures, we don't have a genome for yet. So that's one of the problems. The oldest DNA has been found in ice and permafrost, ranging from 400 to just several months ago, a study that showed 2 million years old of something which we'll look at. But that's in permafrost situations. In the tropics, it's 2 orders of magnitude lower. Heat destroys DNA, okay, which is why we have very little DNA from the hotter African environment. After death, DNA strands are cut into ever smaller fragments with age. And the greatest DNA degrader is water. If water touches DNA, good luck. If it dries out very quickly, you're in much better shape. Bones and teeth are among the best places to find it. In 1990s, Pablo's lab, him basically said, no replicable DNA from ancient amber, and therefore no dinosaur DNA because of the fragmentation that occurs. And he predicted that under optimal conditions, very cold ones, we might be able to get 1 million-year-old DNA. Turns out he was wrong, lucky for us. So originally fresh DNA in your body is one giant long strand. And as it ages after death, it gets smaller and smaller and smaller fragments, basically. And that's the problem, is how do you deal with these very small fragments? It turns out this is the distribution of base pairs in ancient DNA, which means that the vast majority are 50 to 70 base pairs long. And so if you've got a hunk of DNA and it's longer than 70 base pairs, it's not ancient. So it turns out that's helpful in identify what is and isn't ancient. It's simply how long are the fragments. And so ancient DNA is very fragmented, typically 40 to 50 base pairs. It is chemically damaged. Cytosine turns into uracil at the ends of it, and it is mostly non-human in anything you find, basically. Only about 3% turns out to be primate in any ancient DNA you're looking at. It's mostly bacterial contamination. And in the first 12 years of work on ancient DNA, Pablo found human, modern human DNA in every single animal DNA sample he worked with. It turned out the British Museum, Chris Stringer, one of the great paleontologists, his PhD in 1970s was he went around and measured every hominin skull that existed at the time, and his DNA contaminates all of them, okay, as one example. And of course, old paleontologists, to find out whether something was bone, would lick it, okay? And instant DNA contamination by a modern human, all right? So everything that they look at is contaminated. And basically, Pablo and his group became obsessed with how to figure out the contamination issue, basically. Luckily, technology started to step in. And this is the miracle that's helped ancient DNA become a science, basically. And DNA sequencing is the process of determining the base pair sequences in DNA. Any method that's used to determine the order of those 4 bases, basically. PCR, polymerase chain reaction, is the most famous original one, where you could make 1 billion copies of a DNA sample to then look at and analyze. And that's still used when we do COVID testing. We use PCR, basically, to identify what is and isn't COVID. And amplification is this ability to produce millions and billions of copies of the same DNA segment. I love this cartoon. Basically, you're grinding up a bone to put into your computer, so it comes out basically as simply a long list of ACGT, ACG, etc. Okay, and only in 2003 was sequencing ability capable of doing the nuclear DNA as opposed to the mitochondrial DNA. And so research labs for DNA now look like Silicon Valley chip making facilities. Basically ultra clean rooms that have reverse airflow, ultraviolet radiation all of the time when anybody's not there, because even a piece of dust carries DNA. Okay, so you can contaminate any of this stuff routinely, and all of it is contaminated, basically. They've now gotten to the point when they're extracting a bone from a cave site, they wear astronaut suits so they don't contaminate it so much, basically. Um, so it's like a silicon chip factory production to avoid everything. Isolation, high reverse pressure, airflow, ultraviolet light, basically. And you never take DNA from the surface because that's definitively contaminated. You try to get as deep as you can and produce some powder, which you can then extract. So the process is you find yourself a bone, you get it into a clean room, you drill it so that you can produce some powder. You then purify that sample with a variety of chemical methodologies, and then you use these $1 million sequencing machines that are computer driven. Okay. Moore, who just died this week, was the chip manufacturer. He ended up being worth $10 billion. But he came up with Moore's law, which said that silicon chips would basically increase, double their power to compute every 2 years. And the Moore's law of ancient DNA is that the number of studies is doing pretty much the same thing. So this is the published data in 2020, 5,000 research articles, sequenced genomes. Unpublished estimate is that there are 15,000 genomes out there. And everybody is basically going after genome production in every living creature is now what biology is doing. And they all try to eventually compare to a reference genome of some kind that can be updated. So the human reference genome is now GRCH38 is the most current one. It took the first one from 1990 to 2003, it took $2.7 billion to create the first human genome. And since then, sequencing methodology has just been increasing and increasing and increasing and increasing. So that the original Sanger method was very clumsy, and the reads took an immense amount of time. And now you can do massive numbers, billions of reads per hour to do the sequencing ability. This is the latest one, just created last year, called the NovaSeq X series. It can do 26 billion reads per process. It's a 1.2 million machine that are bought by the dozens for anybody doing medical biological research and ancient DNA as well. This is the machine. And you hear about shotgun sequencing, and this is where you randomly break up the genome into small fragments that are sequenced individually. A computer program looks for overlaps in the DNA sequence, using them to reassemble the fragments in their correct order to reconstitute the whole genome. Each nucleotide is identified by a fluorescent tag that the machine can then use to create a particular one of the 4 colors, basically tells you whether it's an A, C, G, or T. Okay, incredible technology, basically. And as a consequence, the number of species that we've done full genomes on continues to expand, basically. 2018, 3,500, 181 plants that they had done. It's up to 1,000 by 1 year later. And now the projection is to try to do all vertebrate species, basically, to get a full genome to look at. And the price of a full genome has basically continued to get lower and lower. Remember, $2 billion for $2 billion for the first human genome. China is aiming to do 1 million genomes. So everybody is in competition now for getting whole genome data on everything that's alive, basically. And there are two companies that now are just advertising for doing your whole genome for $300. Okay, so it's really gone down in terms of price, basically, with these new machines, basically. And the problem, of course, now is they've got massive amounts of data. And so you can no longer be a paleontologist, a bone guy. You have to have a statistician on your team. In fact, many statisticians, because of the sheer amount of data you actually have to collate and compute and do statistical analysis on. So the field has basically shifted toward bioinformationalists. Statisticians are now fundamental to the research. This is an example of the formula for doing the EPAS1 high altitude gene. And I took statistics courses at Berkeley, and none of them covered this kind of advanced statistical computation, basically. So the new rule is kind of grab as much data as you possibly can, hire a great bioinformationalist, and then start asking questions in the resulting data set. So you're basically descriptive looking, as opposed to what we normally think of science as doing a hypothesis testing. It's data mining in terms of producing and describing patterns in the data, but without necessarily doing a null hypothesis kind of way of doing it. So there's been some critique about this, okay? This is the quagga, an extinct creature that they got the first mitochondrial DNA in 1984, okay? So the very first, this is a South African zebra subspecies that died. The last one was in 1883, and they resurrected from 200 base pairs, basically, and showed that DNA survived death, basically, and the very start of paleogenetics. Then Pablo, who in his youth wanted to be an Egyptologist, did his first attempt secretly while he was doing his PhD because his thesis advisor wouldn't agree to do it. But he secretly did the work, and on a 2,400-year-old baby mummy, extracted some DNA and published it in 1985, okay? And of course, years later, he concluded it was totally contaminated, okay? And as a consequence, they became obsessed with defeating contamination in their research, and they have basically created the whole field as a consequence. In 1989, the Taliesin, which is the marsupial wolf of Australia, was studied, and they did 219 base mitochondrial pairs, the second kind of creature that was extracted. Then in 87, a doctoral student named Rebecca Kahn, who was Wilson from UC Berkeley's student, came out with the concept of mitochondrial E, that she did a bunch of comparisons that basically concluded that all modern humans descend from a single African woman and her daughters, and that she lived in Africa somewhere, and about 200K. The range is 99 to 148K now. But that basically proved that we are all African by DNA. This was considered the final nail in the coffin of the original multi-regional theory as opposed to the out of Africa theory. The multi-regional theory said that Homo erectus ended up going into Asia 2 million years ago, and that the Chinese researchers, including some European researchers with them, concluded that those Homo erecti became modern humans in China. All right. And that was the multi-regional theory. And this study basically killed that idea, proving that we originated in Africa, nowhere else. Okay. Now, this is not the first woman. It's not the last common ancestor of Homo sapiens. It is one woman among many who had at least 2 daughters. She's the ancestral genetic phylogenetic estimate. She's a mathematical estimate of how far back current variants of mitochondrial DNA must go in an unbroken daughter, grandmother, et cetera, line to converge on a single individual. And so MT haplogroup is the root of all mitochondrial DNA on the planet. There is also a Y chromosome atom, the man from whom all living humans are patrilineally descended from, who's also around the same time period. Interestingly, he's not the oldest Y chromosome we have. A African-American North Carolina woman sent in her uncle named Perry, Mr. Perry's DNA to Ancestry.com, and they couldn't find a match for his Y chromosome. And they looked at all other labs, and basically they discovered that his Y chromosome matches some in Cameroon, Africa, that's dated to around 350,000 years ago. So there are lots of different, but his Y chromosome wasn't the Y chromosome that ended up populating the world. So again, these are only one woman among many, as he was one man among many, that was the lucky one that ended up having a lot of descendants that lived, basically. Okay. So African lineages are longer and more mutationally diverse because they've had more evolutionary time to do that. And the root of the tree of mankind is African in origin. And later studies have basically continued to say that, in fact, this conclusion was absolutely correct. So it turns out Africa has the richest genetic variation on the planet. And this basically states that your genetic diversity, your heterozygosity, decreases with walking distance from East Africa. So the further you got from Africa all the way down to the tip of South America, you have less and less and less genetic variability. And this is based on the idea of a founder effect, a bottleneck, that the group that moves away always has only a small subset of the total original genetic variability that was Africa, basically. And of course, who do we now currently have the least amount of genetic data from as a country, as a continent? Africa, okay? So we have not done anywhere near the amount of genetic, and we're talking modern humans, not ancient DNA, that we simply don't know. We assume that whites are, if we do the genome from us, that we're okay in terms of thinking about genetic variation, and absolutely not true. So basically the founder effect is the original population, they migrate out, and each of them have slightly different combinations of the original full genetic model. And variability is reduced in each new group produced by this founder effect. And when we talk about I'm blocking on the word I want to use, a bottleneck. This is what we're talking about, basically, the founder effect. This is the Human Odyssey version of this, basically. And that as we moved out of Africa, we basically lost genetic variants, and they estimate that it's somewhere around 50 to 90K ago that this bottleneck occurred. The most recent study I saw was that the current estimate was that about 3,000 to 4,500 people is the group that left, that all of our data is based on, okay? So the rest of the world today has significant founder effects due to this phenomena of leaving a bigger, genetically more variable region. So if you're talking about bottleneck, yes.
I thought the issue with that display was not that we left Africa, but that there was some event that killed off a lot of people.
I think it's actually a founder effect exhibit.
Really?
That's what it's actually showing. And the question is what the bottleneck event actually was. One idea of that whole phenomena is that the bottleneck was simply the founder effect, that a smaller group left Africa and therefore had less genetic variability.
But couldn't the founder effect also be a fact of a smaller group survived, and so their genes were more homogeneous than...
Right. But in some ways, that's the same thing.
The results the same.
Okay, you have to...
The same. I understand the founder effect can be the same thing, but I think that display says it's related to a catastrophe of some kind, not just leaving Africa.
I think that's in there. And the classic explanation, the catastrophe that they reference, is the Mount Toba explosion around 74K in Indonesia, and that created a nuclear winter, and that's been totally disproved.
Okay.
It wasn't that, because they looked at regions in before and after that event, and there's not any change, basically. So they don't think that nobody, as far as I know, usually reference that as why we have this bottleneck. And the more I've read,
I'll look at it again, but if that's the case, if I remember it right, then the display needs to be changed.
Oh, a bunch of the displays need to be changed. Okay, that's why I'm doing these updates.
But Charlie,
every lecture you give, I find I've been saying the wrong things based on the display.
Is it true that the founder effect is for the group that left Africa, but does not apply to Africa, where it has so much more diverse DNA?
Yes.
So
Right.
wasn't it?
But even in Africa, the more we're learning, that's a different lecture. Yeah, I agree with your point. Yeah.
There was a bottleneck. There was a bottleneck.
There was a bottleneck. The question is, what was the bottleneck? Okay. So we go from there. Okay, we'll skip a few of these. But again, not due to the supervolcano Mount Toba explosion in Sumatra in 74K, basically. But the founder is an important point in terms of genetics, because it accounts for why you have more fingers among Amish communities, the Blue people of Kentucky, the presenilin 1 gene in Colombia that creates familial Alzheimer's, where you die at age 50 after 10 years of dementia, the mutiny on the Bounty, Pitcairn Island survivors, the Afrikaners in South Africa, who were a very small founding population that has hugely more Huntington's disease connected to it. One one of the points about this is I just mentioned that the latest update is that they think it was a group of around 3,000 to 4,000 people that exited during this founding event. Okay. And that also implies that we were about the same size at this point as the Neanderthal population in Eurasia, which means we were immensely lucky. Okay, it could have been us that didn't make it just as much as Neanderthals. So we keep commenting about why Neanderthals died out, but in fact, any group like this could have died out in effect. Okay, so we know that. Yeah.
Couldn't more people come out of Africa after we died out or so to keep it
Oh
going?
yeah, no. Eventually, this is the original group.
Okay.
Eventually, more and more and more Africans ended up heading out, basically. But the original population that they think split. Well, as you'll see, this is there's a whole other issue attached to this. Okay, we'll come to it. The first methodological advance was PCR, basically. That was the big start, and it actually won the Nobel Prize in 1983 because it was so important. This is the group that basically started the Human Genome Project. Here is Francis Collins, who eventually headed the project, and here's James Watson, okay, of Crick and Watson, okay. And it turned out at Pablo's lab in Leipzig, Matthias Cringe did the first mitochondrial DNA sequence of a Neanderthal. They just got 300 bases basically out of the Felderhofer Neanderthal #1, part of the femur, basically. His phone message to Pablo was, "It's not human." Okay, that we had in effect two different quote species. So they used the original DNA from one of the femurs, basically, that cut out a segment. And at that point, in terms of technology, they could only extract mitochondrial DNA in 97. They found 27 differences genetically, basically, in this 377 base pair. And their conclusion was that Neanderthal mitochondrial DNA falls outside of the variation of present day modern humans. And this supported the idea then that Neanderthals were simply totally replaced by modern humans, okay? In fact, today, there are no Neanderthal mitochondrial DNA and no Neanderthal Y DNA that we have in any of the data that we have. Okay, we'll come to a potential explanation in a bit. They did the first mouse in 2002. They set up online genome browsers back in 2002. In 2002, they discover a skull in Romania that was dated to 40K called the Awase 1 skull. And in 2015, they showed that he had a great grandfather who was a full Neanderthal. This was a modern human. By the way, I use the term modern human in all my talks, but what I should officially say is African Homo sapiens, because none of these guys were modern, okay? These were older versions of us that we're talking about. So that's the conversion in your head that should happen, okay? Modern humans is just a quicker way to say it. But it turns out they've done the genomes of like 5 different modern human hunter-gatherers in Europe that date to around 40 to 45K. All of them have Neanderthal DNA, and much more Neanderthal DNA than the 2% we have now. So this seems to imply that we were messing around with Neanderthals for a very long time before 40 or 50K. Okay, we'll come to more of that.
So Charlie, modern
yeah.
humans, couple questions, just so I get it clear. They go back how far, and do they have a characteristic DNA profile that covers, is it 70 base pairs or more that flip them over into the modern versus the ancient?
So the oldest skull of modern humans that we got is the Morocco Jebel Irhoud skull that was recently found, that's dated to 315K. The most anatomically modern human skull is Herto and Omo in Eastern Africa that are dated to around 230K. The skull looks much more globular. In fact, it doesn't get totally globular until after 100K. So those are what we consider anatomically modern. The DNA is the same all the way going back. Okay, so your DNA, if we could get the Jebel Irhoud DNA, would probably be a total match. Okay, so we've had pretty much the same DNA from the beginning. It's, in effect, if you look at the reference gene, that's what you should expect to see.
But these modern humans going way back do not have Neanderthal DNA.
They don't have the DNA yet. Okay. We'll be talking. We'll keep talking.
No, they don't have. They would not have Neanderthal DNA, so they would be different from yours and mine, right?
Yeah, if we could get the DNA and they had Neanderthal, it would be different. Yeah.
Yeah. Okay.
Yeah.
So Charlie?
Yes.
Oh, the question I have is, we're saying that in Africa, they have a more diverse DNA because they've been around longer, and the group that left has more restrictive DNA because they didn't mix as well. But if people constantly came out of Africa, they would have mixed with the people that were already there. And also people in Africa don't really mix that much because they lived in small groups. And, unless they move from one part of Africa, south to north or whatever, they're not really mixing. So how do they explain the difference in the diversity of the DNA in that situation?
Well, in fact, as we'll eventually see, there was a lot of mixing in Africa going on. Okay. So they're going to be slightly different founder effects even within Africa. Okay, so lots of mixing, and if you have a particular mutation in one group and you mix, you may or may not end up transferring that to another group. So it's recombination again. That's the issue.
But how continuously did people leave Africa to go to quote
always,
the New World or whatever?
Yeah,
always.
probably as long as there was game moving in that direction, they'd follow it. And this is all walking. Basically, they estimate it only took less than 20,000 years to get from Africa to the end of China. If you're just walking, following herds, basically. They do the rice genome. And this was the last time they used Sanger sequencing. 2005, they do next generation sequencing and upgrade faster processing of information. When they did the chimpanzee DNA, they did 2,000 million bases basically at this point, and we have a 1.06 difference from chimp DNA basically. So about 90, they quote 98, 99% difference, okay? Slightly less than with Neanderthals, and they have one chromosomal fusion that we don't have. And 29% of all the proteins were identical between the two of us, even though we split 7 million years ago. By the way, the fun fact on the floor I use is I show them the chimp skull, and I say, how many fossils do we have of chimpanzees who have been on the planet for 7 million years? You know the answer to that? We have 5 teeth, period. And the explanation is chimpanzees and gorillas live in rainforests, and rainforest floors have humic acid in it, which eats bone. And as a consequence, we have no fossil bones of chimpanzees in our entire collection, okay? Fun fact. 2006, they do a partial sequence of the Neanderthal genome. And then in 2007, they find some Neanderthals living in Siberia, okay, Denisova cave. They find that they have a variant for a red hair gene and a fair skin gene in Neanderthals, and that Neanderthals have a version of the FOXP2 language gene that's almost identical to ours, okay? I believe clearly Neanderthals were capable of communicating. What that consisted of is still the big debate, and we may never actually know. But both the hyoid bone is identical to ours, and that's what holds our tongue and ability to make certain kinds of sounds, etc. It may be that they actually had a higher pitched voice based on the location of the hyoid bone in them. In 2007, they figured out how to do sequencing of proteins that were regulating gene expression. And this was the beginning of the epigenetic revolution. And basically, remember that you can get chemical markers via your experience in life that will change the expression of your DNA. It won't change your DNA, but it'll change your expression of it. And that they discovered that this was transgenerational, non-Darwinian, Lamarckian kind of, if you have an experience in this life, your grandkids will suffer in some way because of what you did. Okay. The original data was the famous winter starvation period in Holland in 1944, where half of the country starved for the entire winter, basically. And they've shown effects in grandchildren of these people in terms of obesity rates, psychiatric syndromes, etc. Holocaust survivors who were starved and had PTSD have epigenetic changes. And just within the month, they changed the epigenetic chromatin thing that controls expression, which produces fat mice, and showed that that chromatin epigenetic change lasted for the next 4 generations. Okay, so that they've now absolutely proved in my mind that epigenetic changes are transgenerational. Okay, so don't starve or your grandchildren are going to pay for it, basically. Okay, is the consequence. This is an amazing kind of data that keeps coming up.
Say, Charlie, you mean that the grandchildren would be obese, or would they be thin?
Right, they all they turned out to be, in this case with the mice, obesity was the thing that got changed by the epigenetic change.
And that happens to humans
think of all the amount of structural racism in this country. And recently they showed that any kind of chronic stress by anybody increases the amount of, I'm blanking on the chemical in your brain that basically kills off hippocampal cells. Okay, so the more stress, the more chance that you're going to get demented in old age, and that potentially these kinds of epigenetic stress-related changes can be transgenerational. Okay. So
Oh,
this
one
is,
last. Mhm.
go ahead.
I forgot about when you were talking about chimpanzees. Do we have any skulls or bones of gorillas, or is it just teeth?
Just teeth for
Just
both
teeth of gorillas
gorillas,
too.
and teeth for gorillas.
Oh.
Bernard Wood thinks that we may simply have not searched well enough. Okay, that there may be some bones out there, but we just haven't found them. Okay, but most bones do get disintegrated by the humic acid is the problem. So this is your DNA strand basically, and genes are surrounded by, in effect, what turns out to be a kind of string that either tightens or lets go. In the tighten phrase, your DNA gets turned off. When it expands, it turns on. So this is how epigenetics can control your gene expression, basically. So tight, no activation, widening, and you get some activation, basically. So it depends on your wrap. And this is what changes in terms of the epigenetics. Okay, let's see. They've shown that there were multiple lineages of Neanderthals, basically. There wasn't just one genetic type in Eastern, Western, Southern Eurasia. And they've basically shown, we'll come to it. And the big finding has been repeated that Neanderthal mitochondrial DNA differ from each other by about 20 bases and are only 1/3 as diverse as modern humans. The big finding has repeatedly been shown that Neanderthal DNA is genetically very, very unvariable. That the equivalent today is mountain gorillas, who are an endangered species because of the low number of gorillas that are present. And that basically the genes indicate that Neanderthals had a small population size. Churchill called it, they were thin on the ground, basically. And part of this was the repeated glaciation periods, warming periods in Europe and Eurasia, where basically the glaciation destroyed ecological fauna, plants, etc., and crashed Neanderthal populations. And then they basically had to rebuild once it began getting warmer. But this kept happening over and over and over again, which reduced the genetic variability in Neanderthal, so that lots of people today believe that that was probably the main reason why Neanderthals disappeared, not us. They would have gone extinct anyway, basically, is the thinking because of this lack of genetic variability. So they do, many people think of the beginning of ancient DNA as the first draft of the Neanderthal genome that happened in 2010. They then, in the same year, publish the little pinky bone results. They destroyed the pinky bone in their attempt to get the DNA, but it basically proved, these two prove, today the numbers to quote are that there's 1 to 2% Neanderthal DNA, not 1 to 4, which was the original estimate. So that's the amount of DNA in modern humans. There's 5 to 6% Denisovan DNA in Melanesians and Papua New Guineans. And there's a 0.2% Denisovan and Neanderthal DNA in Asians and Native Americans. And because of black backflow of modern humans into Africa, there is 0.3% Neanderthal DNA in Africans today. Okay? So everybody in the world today has Neanderthal DNA. Even Africans, the San of the Kalahari, actually have a relatively high Eurasian DNA count because of all the admixtures that have happened there. Okay, so Pablo gets the credit for the push to get Neanderthal genome, and he got it basically. Okay, and the two publications that created the field were the draft sequence of the Neanderthal DNA, not the full genome, but the draft, as well as the Denisovan DNA that was gotten from the little girl's pinky bone that was found in Denisova Cave. And the Neanderthal one was based on these three bones from a cave in Croatia, Vindinja. And they got about 55% of the Neanderthal genome, basically. This bone was dumped in a box full of animal bones because that's what they thought it was. And Denisova Cave has all of these, in effect, less than 1 inch pieces of bones by the thousands that they're systematically going through to find whether they were human or faunal, etc. That research continues. And this draft sequence was done by Richard Green, who was one of Pablo's group in Leipzig. He's at UC Santa Cruz these days. But he's the one that quoted the 1 to 4%, which is no longer, you shouldn't be telling people that, it's 1 to 2%, basically. Now, so we discover that Neanderthal is 99.7% identical. Okay, so we are basically the same creature as Neanderthals. That compares to 98.8% with chimpanzee DNA, and that there are 9 million base pair differences between Neanderthal and modern humans. And so 1 to 2% is what you should be quoting. That's the same amount as you would inherit from a 4th great-grandparent. Okay, and then they figured out the split via molecular mitochondrial DNA clock, that we split from Neanderthal around 760 to 550,000 years ago. So we've both been around since around this period. The oldest DNA that we have for Neanderthal is now at 430K from Cima de los Huesos. Now, interestingly, there's incredible little difference, basically, between the two of us. That said, interestingly, Pablo and his Leipzig group have refused to give a classic Latin binomial name to the Denisovans. They refused. Yes? Okay. So no Homo denisova or denisovensis, okay? They basically believe, they don't believe in species, all right? And that the Russian collaborators use the term Homo altaiensis from the Altai Mountains where Denisova is, basically. And Pablo now recommends against imagining separate species of human evolution, not us versus them, but one enormous metapopulation composed of shifting clusters of essentially humanish things that periodically coincide in time and space, and when they happen to bump into each other, they occasionally have sex. Finlayson, the Gibraltar Neanderthal specialist, says each valley could have told a different story. In one, they may have hit each other over the head. In another, they may have made love. In another, they ignored each other. And Moel Allen says a super long elevator ride with strangers. Okay, I like that one. In 2013, the third oldest DNA was pulled out of a horse femur from the Yukon region of Canada, dated to around 700,000 years ago, basically. And the DNA sequence from this horse, okay? And 2012, we get the full nuclear sequence from the Denisova genome. And we then get a year later the mitochondrial genome sequence from the Cima de los Huesos 28 skeletons, basically. And amazingly, the mitochondrial sequence turns out to be Denisovan. The same year, they do the nuclear DNA, and that turns out to be Neanderthal. Okay, so and there are multiple competing interpretations of how this was possible, that you could get mitochondrial DNA. If we have enough time today, I'll get to one possible explanation, basically. But out of the femur, they got the mitochondrial DNA that said they were closer to Denisovans than to Neanderthals. Lots of explanations. Let's see. If you look at what differences there are between humans and all of our nearest extinct relatives, in 3,000 base pairs, there are 31,000 single base pair differences, 4,000 insertions and deletions. There are 100,000 substitutions. There are 87 genes. Very small amount make for some interesting differences, basically. And as I mentioned, there was not just one population of Neanderthals, but multiple populations. One is considered the Altai Denisova Neanderthal, which was the furthest east that Neanderthals got. We don't actually know that. We simply haven't found any Neanderthals further, as an example, into China. There may be Chinese Neanderthals for all we know. They had a massive territory, okay? At least 4,500 miles between Wales and Denisova that they basically were in. And some people actually think Neanderthals were an Eastern group that went into Europe, okay, as opposed to the reverse. Lots of admixture going on, basically. A revision of the 31,000 is now that there are 12,000 basically differences, so less than we thought before. And this involves things like cell division, brain growth, the cellular features of neurons, some craniofacial phenotype stuff. Pablo says the dirty little secret of genomics is that we know next to nothing about how a genome or a gene translates into the particularities of a living, breathing individual. Okay? These are the 96 human-specific amino acids that we know of. Only humans have these particular amino acids. And there are what are called human accelerated regions that separate us from chimpanzees. They are mostly regulatory, not gene differences in about 118 bases, mostly involved with cortical development that made us us rather than a chimpanzee, but in the non-coding regions, basically. So this is the classic example of some of the, it's very clear that different hominin species, when they met up, like to have sex together. That's the basic data, okay? And so Denisovans mixed it up with us, Neanderthals mixed it up with us and Denisovans. An unknown, probably Homo erectus, got some material into the Denisovans, which then got into us. So these are the current numbers that you should be quoting if you quote that 1.5 to 2.1 Neanderthal DNA in modern humans. There's 0.5 Neanderthal into Denisovans. There is 3 to 6% Denisovan into modern humans, mostly in Indonesia. And there's this unknown hominin, which most people think is Homo erectus, that got into the Denisovans as well. Okay, and that's pretty much the same thing. And that Neanderthals and modern Africans are around 0.3%. And you have Neanderthal and Denisovan in Native Americans, that is at the 0.2% level. Now, East Asians carry more Neanderthal DNA than Western European types do. Okay, slightly more. They think possibly because there was more admixture in East Asia. It's possible one explanation. So all modern non-Africans are estimated to carry 2 to 8% archaic human sequence of both Neanderthal and Denisovan variety. So we know that Neanderthals were heavily interbred. The Denisova Neanderthals were related at the half-sibling level, lots of mixing, and very low effective population sizes, possibly by an order of magnitude lower than early anatomical modern. And some estimates are that the genetic fitness, because of all of this, was as much as 40% less than modern humans at the same time. So they were, in effect, a population on the way out genetically if something didn't happen. And it may be that we come in using the same faunal population hunting sources, and that may have been enough to shift the burden, but there are some other explanations as well. Why was Neanderthal DNA replaced in modern humans once it got in? Because of the small population size, there were Neanderthals had more deleterious variants, because if you have small population groups, that's where you get deleterious DNA. But some Neanderthal DNA fixated because they were actually more functional than the modern human version, okay? So there are estimates basically about, again, the number of Neanderthals at around 40K when they disappear. And this is probably the best estimate, is that there were about 1,000 to 5,000 Neanderthals on the planet at the time when they disappeared. A later estimate has been discredited. Note that we may not have been much bigger at the same time, all right? But that's the rate, and this was potentially far too genetically possible to survive in the long run, basically. I had a visitor at the academy asking me if we're 99.7% identical, how can modern humans have 2% Neanderthal DNA per Ancestry.com? The correct response is that the 99.7% identical between us refers to all 3 billion base pairs, which leads to a 0.3% base pair difference between the two of us. You have approximately 2% Neanderthal DNA, but the 2% refers to the 2% of that 0.3% base pair difference. So there's only about 180 100,000 Neanderthal base pairs that exist in us. And that's what we're talking about. Interestingly, the estimate is that there is massively more Neanderthal DNA in humans today than there was while they were in their entire history on the planet. Okay, so they survived in us, basically. We'll skip that. They estimate that in current humans, 40 or so % of the entire Neanderthal genome still exists. And that, but to remember that in each of us, my DNA is different than your Neanderthal DNA. We don't all have the same Neanderthal genes, basically. And there's about 10% of the entire Denisovan genome that's in modern humans. I've never seen an explanation of why the difference, but that's the estimate currently. And incredibly low genetic variability among Neanderthals. These are modern Africans, modern non-Africans, and this is the amount of DNA variability, basically, in Neanderthals. So much lower than present day humans. In fact, it's among the lowest measured for any biological organism that has survived. And the reduction in population size began a long time ago for the Neanderthals, again, because of these probably repeated population crashes and the fact that they tended to have communal population sizes of about 20 to 25 family groups that lived in separate valleys that occasionally got together, which made it harder. Some people think that there was better social communication among modern humans coming into Europe compared to the kind of connections that Neanderthals had. And one reason for the Neanderthal demise idea is that this low population number with interbreeding basically would have crashed them independent of us ever being there, basically. So while only 1 to 2% of the total genome of moderns is Neanderthal, this represents about 40% of the total Neanderthal genome. That's something you can quote that people find interesting. And again, there's more Neanderthal in Eastern groups than there is in Europe, okay? Maybe because this group's had more intermixing than the groups over here. We're not sure, basically. And the, again, this OASI-1 Romanian skeleton proved to be very important because he had about 6 to 9%, maybe 10% Neanderthal DNA in him at 42K, which represented 4 to 6 generations back. He had a full Neanderthal grandfather. And so if you take generation 0 as being 100% human, then the hybrid would be 50% Neanderthal, 50% human. And by the way, the fact that we have this 2% says that all of these intermixing produce hybrids that were well taken care of enough to produce more hybrids, okay, in terms of how much of a difference there was between us, in terms of looks and behaviors, et cetera. They took care of their hybrids, basically, as if they were the same. But each generation, you get less and less Neanderthal DNA to, at 5 generations, you're basically down to about 3%. The big question then is, why didn't this 2% disappear in us?
Right.
Okay. And the reason is adaptive integration. That is that these gene Neanderthal variants did something good for us and that were better for us in Eurasia than our original African version of those genes. Okay, so they turned out to be helpful, is why they got kept by selective natural selection, basically. And as I've mentioned, those early hominins coming into Eastern Europe at around 45 to 40K appeared to have, Pablo thinks they mixed quite happily, extensively, and frequently. And as a consequence, all of the skeletons that we discovered already have Neanderthal DNA in them. The cave where they find Oasi 1 and also the Bakokiro cave in Bulgaria, that you can see at different times in this particular cave that the Neanderthal DNA amounts in the skeleton get less and less, but starting from more than we have today. So if you start out with 10% after an admixture, it eventually gets selected out down to the 2% that we have today. And so this is the Oasi 1, who's got, at this point, has 3% Neanderthal DNA. And this is where they lie on various chromosomes. And note, some areas are massively Neanderthal and some are deserts. Okay, that's an important fact. And basically from, let's say, 10% in Oasi, we've gone down to about the present 2% of Neanderthal. And these are the 5 40 to 45K modern humans that basically showed up early. Interestingly, none of these left descendants. Okay, so they didn't make it, which again says that we were lucky in making it, because many of them, local modern human groups, didn't make it, basically. But basically, the longer we've been around, the less amount of Neanderthal DNA shows up in our system. We'll never know whether sex between Neanderthals and modern humans were kidnapping or romance, basically, quest for fire or dancing with wolves kind of situation. Lots of Neanderthal specialists think they were similar enough that it was probably an okay engagement, basically. Okay, let's see. We've said some of this already. Natural selection or negative selection is basically going to get rid of any variant that doesn't work for you or that could potentially harm you. And so there are large regions in modern humans with strongly reduced Neanderthal DNA due to negative selection. And the most pronounced happen to be on the Y and the X chromosome. There's almost no Neanderthal in those. And also in most conserved, functionally important modern human gene areas, there is no DNA that's Neanderthal. Okay, so almost all of Neanderthal DNA in us is in regulatory junk DNA regions. Okay. Okay. Part of the DNA has resolved some famous controversies. Kennewick man found in Washington was an 8,000-year-old skeleton found in 1996. His skull, though, looked to many people to be like the Anhui in Japan, the ancient group, so that they looked more Polynesian than Native American. But they finally do the DNA, and in fact, they are totally Native American genetics that they find. The big issue, one issue here is that we treated Native Americans horrifically when we initially started getting DNA from North American groups of tribes, saying that we're using it for diabetes research, and then we do early origin research, which goes against their entire religious conception of themselves. Okay, and this was the battle here. Ultimately, they reburied Kennewick Man in now a hidden grave, basically. So they then, in 2014, find what's known as the Anzic, Montana infant that's buried with Clovis tools, which was initially considered the first group to come into the Americas. And in Siberia, there's a famous 23,000-year-old boy that's called the Malta child that was sequenced. And he was more closely related to Europeans and Native Americans more than he was to the local Siberians that existed now. And it turns out he was a representative of the Amnaya group that entered Europe, which is why he's more closely related to Europeans and also related to Siberians that ultimately went into North America. And there's the Native American connection. Eventually, they do studies of Native Americans not in the United States, but in Canada and in all of South America. We've got very poor representation because of the bad way we treated Native Americans in the United States. You want to be sensitive about genetics and Native Americans, but basically their origin stories are basically like Christian fundamentalists who believe that science doesn't tell them anything. And it's basically totally hindered genetic studies of United States Native Americans as a consequence. It's getting a little bit better, but still a lot of damage was done there. In 2016, we discovered that the Neanderthals in Cima de los Huesos were 430,000 years ago. They've since found that there are at least 3 different genetic types of Denisovans. One maybe needs a new alternate species name because they were much more different than the other 2. And the current thinking is that about 1.5 to 7% contains uniquely human DNA. Let's see. There's the famous comment about the fact that 2 humans on separate continents are closer genetically than 2 chimpanzees on the opposite side of the Congo River. Okay, that again, chimpanzees have massively more genetic diversity than all 7 billion modern humans, basically. So that it points out that genetic diversity in non-Africans is actually incredibly low compared to the African genomes. This was an interesting study because what it showed was if you take each gene and look at how far back it created a common ancestor, the most recent shared ancestor for modern humans is around 320,000 years ago. Interestingly, just around the same time as the Jebel Irhoud Moroccan skull that we have. So that the most recent common ancestor for all present day people is in effect around 320K. This totally disproves Richard Klein's theory of that we had a genetic brain change at 50K, okay? because this data totally does not support that idea. Our most recent ancestor somewhere popped up around 300,000 years ago. And that's an important little piece. And again, in terms of genealogy, you get more and more ancestors going back. So eventually your paper trail is going to contain way less than the actual number of people you had back in your genealogy. The debate, let me think if, yeah, too much information. We may have to next time do more of this and save the Neanderthals to the following session or so. But Chris Stringer, a British museum, Milford Volpoff from Michigan, had this 30-year battle basically about multi-regionalism versus out of Africa ideas. Basically that multi-regionalism said we met and mingled and had offspring locally, and so produced Homo sapiens out of Homo erecti. Their battle was long and debated, but eventually with DNA you can actually start figuring it out, and basically the DNA, mitochondrial Eve, et cetera, says that it's out of Africa that wins this debate. Pablo thinks that the best current model is that we replaced Neanderthals with hybridization. Fred Smith thinks that we actually assimilated. We totally mixed with Neanderthals, and that's the way they disappeared. And these are probably the two leading theories about what happened to the Neanderthals, even though, as I've said, the genetic variability in Neanderthals was so low that they were on their way out anyway.
Yes.
But mitochondrial Eve basically was the death knell for multi-regionalism, except that the concept of multi-regionalism has now popped its head up again in terms of Chris Stringer now believes that Homo heidelbergensis in the original theory was the ancestor of both modern humans and Neanderthals. Some of the redating of skulls in Africa, like the Broken Hill Kawabe skull, this beautiful skull, was redated to around 300K.
Right?
Somebody is talking back there.
Thank you. See you then. No problem. See you Wednesday.
Okay. So some people are now thinking that Homo heidelbergensis only was the ancestor of Neanderthals, not of modern humans. And that modern humans came about through a mixture of a variety of groups in Africa that eventually commingled and admixed enough DNA so that we got the final globular skull version of anatomically modern humans, basically. That's probably the leading theory that's being talked about out there at this point. Let's see, that basically is the gist of that report. And that a simulation may have been. There are people who don't accept that, but it's one of the ones that are in contention, basically. In 2021, they discovered some mammoth teeth that puts DNA at 1.6 million years ago. The oldest DNA that was found at that time, a woolly mammoth. And in 2021, they showed that Neanderthals had basically the same blood types as we did. Again, this confirmed African origin, low genetic variability, weak fertility, and more viral infections that lead to high infant mortality rates in Neanderthals. Again, another bad news kind of finding. And then the most spectacular study of this year is 15 years ago, I always get his name wrong, Wildershlev, the Danish paleontologist, came upon this Arctic desert in Greenland that was underwater for a pretty good period of time. But today it's the home of moss, lichen, and muskox, basically. Terrible Arctic desert, basically. But it preserved some very old sediment, basically. Cap Copenhagen Formation is what it's called. And they ended up digging 5 shovelfuls of sediment 15 years ago, and it took them 15 years for the technology to blossom for them to finally extract the DNA from this 2 million year old site. And what they proved was this, that 2 million years ago, this is what it looked like. Basically, popular birch forests with mastodons, rabbits, birds, etc. So they got 135 different DNA-based species, 100 plant genera, 9 different animal taxa, including caribou, hares, lemmings, rodents, geese, fleas, ants, crab, coral DNA, proving that it was a much warmer period during that time. And the way they did this was, it turns out that because of the marine deposition of this stuff, it favored the absorption of DNA by mineral surfaces on clay and quartz that somehow preserve the DNA. So it chemically bonded to the minerals and preserved it. And basically, they're now working on a 4 million year old sediment to see if DNA was possibly preserved that long. So that's DNA. And the protein analysis, basically using what's preserved of collagen from bones, that protein, they have basically found collagen from a 3.4 million year old camel bone, some eggshells in Central Africa that are 3.8 million year proteins. And amazingly, they have found a 195 million year old protein from a herbivore dinosaur, as well as another 2 million year old rhinoceros. So proteins last way longer, okay? And I think we have reached the bewitching hour. There's just way too much material. So if you'll indulge me, next month we will finish Neanderthal and add Denisovans as one package, and then finally the following session we'll do the renovation of Neanderthals, basically about all of the incredible new data that makes Neanderthals look a whole lot different than the cavemen we used to think of. Okay, so thanks for being generous with your time.
What's the date and time again for next time?
So again, it'll be the last Monday of April. Okay, whatever that date is. Let's see.
Looks like that's the 24th.
The 24th. Okay.
1 to
So
3.
one to three, basically. We will continue on this ancient DNA stuff. All right. Thanks for being patient. Have
Hey,
fun
Charlie?
with.
Thank you,
Yes,
Charlie.
Dennis.
Thank you.
One
Okay.
quick question about the, you're saying that none of the harmful genes have come through to us from the Neanderthals. I thought they had some
No,
genes.
no,
that
that's not
affect our
true.
diabetes. Oh, that's not true. Okay.
No.
We didn't get that far.
Oh, we didn't get that far. Okay.
Well, next time we'll talk about what functional Neanderthal DNA there is in us that actually both helps us and hurts us.
Haplogroups, yes.
And one last
Yeah.
quick question about genes and stuff. Do we have any that go back to other species we came from? I mean, like they say birds came from dinosaurs or
Right.
related to
No,
dinosaurs.
the
No.
oldest hominin DNA is the Simo de los Huesos Neanderthal from 430K. That's the oldest hominin DNA.
Okay.
Hopefully that we keep wanting to find a Homo erectus or a Neanderthal that was preserved in permafrost. That would be a spectacular find. Okay. So, and maybe it will happen. They found mammoths, etc., that are perfectly preserved. Okay. So see you next time. Okay.
Okay.
All
Have
right,
fun.
thank you.
All right. Take care.
Thank you, Charlie.
Bye-bye.